Hello, class. We are now going to go into chapter 20, talk about the lymphatic and the immune system, talk about how our body deals with cell damage, how our body deals with pathogens, intruders to the body, how our body prevents intruders from coming into the body, potential intruders from coming in, and that whole process. The immune and lymphatic systems work together, what we call immunity, right? It protects the body from cellular injury and disease causing cells known as pathogens. The immune system tries to defend our body against internal cancer, can develop internally, and external threats, viruses, bacteria coming from outside the body. Immune system doesn't have organs, but consists of cells and proteins. We're going to talk about the different cells and proteins, the white blood cells, for instance, the leukocytes, the different proteins, the complement proteins, the that are that help with immunity. We also have the lymphatic system, which does contain organs and tissues, which help uh, remove pathogens, but also with the lymph nodes, the spleen, and the thymus, have also have other functions in maintaining our fluid balance and our fluid homeostasis. So make sure we have enough fluid in the body. So not only do the kidneys help make sure we have enough fluid in our bloodstream, but also the lymphatic system as well. Basic functions include regulation of interstitial fluid volume, so the fluid that exits the capillaries. We have to get that fluid out of the interstitial extracellular space, transport it, and deliver it back into the cardiovascular system. When that fluid enters the lymphatic vessels, it's called lymph. Also, we have the absorption of dietary fats that can too large to pass between endothelial cells of blood capillaries. Remember, the capillaries, there's different type of blood capillaries. Some are a little tighter, less leaky, and then you have some that are more leaky and very leaky. So some of these fats cannot pass through the endothelial cells of the blood capillaries. And so they enter the lymphatic vessels in the small intestines. So the absorption of dietary fats. And then we have, like we talked about on the previous slide, the immune functions of the lymphoid organs, able to filter pathogens from your bloodstream. Right, this is showing you the lymphatic system, lymphatic vessels. We have tonsils, which we're going to discuss, called MALT. We have the thymus, makes T cells. We have lymph nodes, which can trap pathogens. We have the spleen, the largest organ in the lymphatic system. Also can, plays a role in our body getting rid of old red blood cells, but also getting rid of pathogens. Large intestines, small intestines, stomach, you see that there. Lymph is collected in vessels called lymph collecting vessels, merge to form lymph trunks, right? Similar to how the flow of blood is in the body, we have sort of like the flow of lymph. Remember, the lymph is fluid that has come from the bloodstream the blood plasma that has exited the bloodstream, entered the interstitial spaces, entered the tissues, is picked up by the lymphatic vessels, and then is eventually transported back into the bloodstream via the venular, via the veins. So we have similar structure to the arteries and veins. These lymph trunks, intestinal trunk, lumbar trunk, drain into a large swollen vessel called a cisterna chyli. Remaining trunks drain into the right lymphatic duct, drains into a junction, the right internal jugular and right subclavian veins, drains upper right side of the body, right? And so we see this lymphatic system, the green, and it drains back into uh, the vein, the brachiocephalic vein here. We see, and re-enters the bloodstream. So the fluid that is lost is then re-entered into the bloodstream. Remember, we're going to talk about it's gone through some lymph nodes and some processing along the way so that pathogens have been cleared. Maybe not all pathogens, but many pathogens have been cleared from it. The lymphatic vessels make up a low pressure circuit. There's no main pump, there's no heart pumping the fluid in the lymphatic vessels. Most of them are traveling against gravity. Similar to veins, they have valves that prevent it from flowing backwards. They're lodged in between muscles. So as you move your muscles, which exercise, movement is good for the body, but movement of those muscles causes movement of this lymph fluid. You also have some contractions of smooth muscles found in the walls of lymph collecting vessels, and that's how the lymph is able to move. We have lymphatic capillaries, which are around the arterial capillaries, the capillaries of the systemic system, the systemic blood flow. And so as blood is released from the capillaries and as fluid is released from the capillaries um, into the interstitial space, is collected in lymphatic capillaries. We don't want fluid flowing out of the lymphatic capillaries back into the interstitial space, so that it's a one-way movement, which is different from the capillaries in our bloodstream, because we know that the capillaries in our bloodstream, closer to the arterial end, we have fluid going out, but closer to the venual end, we have fluid coming in. So some of the fluid comes back in at the venous end of our capillaries. Some of the fluid enters the lymphatic system and becomes what we call lymph. Okay? Same type of fluid, but goes just to different places. And so this is showing us an arterial, interstitial fluid, the lymphatic capillaries, blood capillaries, all kind of intertwined together, the venual, right? And so as fluid enters the capillary, enters the tissue cells, the interstitial space, the space between the cells of this tissue, the more fluid that's there, the more pressure that's increased. As the pressure increases, it these um, opens up these gates so that fluid now flows into the lymphatic vessels, the lymphatic capillaries, and eventually, and then we call it lymph, and it flows into our lymph system, eventually returning to the venous to the veins later on down the line. We've already mentioned edema in this class, but it is swelling, excess interstitial fluid, fluid in between the cells of a tissue, interstitial fluid, uh, and many things can cause this to happen. Lymphedema is generally due to removal of lymphatic vessels during surgery or blockage of vessels from pathogens such as parasites. So if fluid can't get into your lymphatic system or gets blocked from getting in, or lymphatic vessels get destroyed during surgery, you can have lymphedema. Because where is that fluid then going to go? It's going to start swelling, and you get what is called lymphedema. And fluid accumulates in the tissues. Your The predominant tissue type of lymphatic system is this loose connective tissue called reticular tissue. Um, if you think of reticular tissue, think of like a web-like tissue, a net-like tissue designed functionally, anatomically, to be able to trap disease-causing pathogens. Lymphatic reticular tissue is typically called lymphoid tissue and is found in the lymphoid organs. And you have varying degrees of organization. Lymphoid organs also house leukocytes. Leukocytes, you may have remembered those when we were talking about red blood cells and white blood cells. But we have different types of leukocytes. Macrophages, which are active phagocytes, ingest harmful pathogens and destroy them. We have B and T lymphocytes. A, A granulocytes means they don't have granules. They function in innate immunity as well as in learned immunity. Dendritic cells, think of dendrites projections. So we have spiny processes resembling dendrites of the neurons. Reticular cells, particularly abundant in the spleen and lymph nodes, and they produce reticular fibers. Right, so those are the tissues, lymphoid tissues and organs. This is the cells that are in the um, reticular tissue. I mentioned this a little bit earlier, but we have mucosa-associated lymphatic tissue, MALT, loosely organized clusters of lymphoid tissue, which protects mucous membranes, exposed to large numbers of pathogens. So what part of the body do you think will be exposed to large numbers of pathogens? Well, any part of your body that is open to the outside. So coming in we have to breathe, so things can come in our nose. It's like an open door to the outside pathogens. The mouth as well. We're eating. Unfortunately, people put their fingers and their hands in their mouth. We're breathing around other people who may be sneezing or coughing, or just germs floating around in the air, bacteria, viruses can come in through the mouth. So, where things could potentially enter is where are good places for us to have what is called MALT, mucosa associated lymphatic tissue, right? So, specialized MALT is found in 3 locations: tonsils, around the oral and nasal cavities, right? And there's 3 different types of tonsils located there. Posterior nasal cavity, posterior nasal cavity, so the back of the nasal cavity, posterolateral oral cavity, and the base of the tongue and the mouth. We also have Peyer's patches uh, in the small intestines. Things that we eat can oftentimes contain pathogens. And then we also have the appendix. Also, things that come into our body via the mouth. Eventually, could get to the large intestines, and we have the appendix, which could defend the body from bacteria in large intestines. Right, so those are uh, areas of specialized malt. Right, another picture, and the intestines, large intestines, small intestines. Right, to fight off pathogens. Lymph nodes. Sure, everybody's probably heard of these. Small, kind of bean-shaped lymphatic tissue associated along the lymphatic vessels throughout the body. Axillary lymph nodes, cervical lymph nodes in the neck, inguinal in the groin, mesenteric in the abdominal cavity, right? Lymph nodes, lymph flows into these nodes and goes through this reticular network, this reticular tissue. Pathogens get trapped, and then leukocytes, dendritic cells, eliminate the threat, right? So it's like, uh, you know, pathogens enter our body, get into the lymph, which is, remember, fluid that has exited the bloodstream, travels into this, through this lymphatic vessel, into the lymph node, gets trapped in there. Leukocytes, dendritic cells come and destroy it, right? So that it doesn't go to the rest, doesn't get into the rest of our body. See this note here, lymph nodes trap approximately 90% of pathogens in the lymph, prevents them from being delivered to the blood and other tissues and organs, right? So that's our lymphatic system helping us in our immunity, helping prevent disease. Okay, showing you the structure, lymphatic system, lymph nodes, and then showing you a more detail, a lymph node. Lymph coming in through the afferent and out through the efferent. Spleen is the largest lymphoid organ in the body, located on the lateral side, away from the midline, lateral side of the left upper quadrant of the abdominal pelvic cavity. Purplish brown, by the size of a bar of soap. Internal structure consists of reticular fibers made by reticular cells. We have this red pulp of the spleen, which destroys old erythrocytes. Remember, blood sites live about 100 to 120 days. And then they get old, they get beat up, they come to the spleen and get destroyed. Some of them gets recycled. And then we have the white pulp, which filters pathogens from blood and contains leukocytes and dendritic cells. So think of all these different ways so far. We haven't even gotten much past the surface, but pathogens come into the body. If it gets into the lymph, it gets filtered by the lymph nodes, caught in reticular fiber there, attacked by the leukocytes. It could get caught in the malt, the tonsils. It could get caught in the appendix. It could get caught in the spleen. All these different areas of the body that could attack and prevent the pathogen from spreading over into other parts of the body. That's if it gets in the body. Remember that we have skin that prevents quite a bit from getting into the body. Right? So that's the spleen. We also have the thymus, which doesn't trap pathogens, but secretes hormones that enable it to carry out its primary function, which generates T cells, functional T cells, which help protect the body from pathogens. The thymus is larger when you're younger, gets smaller as you get older. less productive as you get older. Um, it has an outer cortex and an inner medulla. Cortex contains densely packed T cells, right? And we're going to talk about what those T cells do later on in this chapter. And that's the thymus, right? So the overview of the immune system, we have three lines of defense against pathogens, and we kind of briefly mentioned them, but goes into a little bit more detail, and we'll get even more detail. So our first line of defense is cutaneous, your skin, mucous membranes that trap pathogens that would or block pathogens from entering the body. Our second line of defense is cells, response cells, and proteins that make up innate immunity, sort of your just quick response, your not non-specific response. And then we have our third line of defense, which is our adaptive immunity. So once we've seen this particular intruder before, our body can adapt to it and have a response specific for that intruder. Right? So these are three lines of defense against pathogens. Our bodies are amazing. And the ability to be able to defend itself. If you think of like an old back in the day when they had castles and they had the wall, then they got the shooters shooting bow and arrows from the top of the wall, and then they got the people throwing, with the catapult and the big fiery bomb thing over the wall. And then you got the troops on the ground that'll come out and get you too. We have multiple lines of defense, makes it extremely difficult for pathogens to get in. Now, obviously, we do have pathogens that get in. We do get sick, get infections, but most of what tries to get in our body gets stopped. And if it does get in, it gets killed. If we have a healthy immune system, which is something that you can continue to work on by not smoking, not drinking, taking care of your body, making sure you have the proper fluids, exercise, eat right, get rest, make sure your body is functioning at its highest level. All right, so immunity is classified according to the way it responds to different pathogens or forms of cellular injury. So you have an innate non-specific immunity, which responds to everything. So we're going to talk about those antimicrobial proteins in certain cells that respond quickly within the first 12 hours. Then we also have adaptive immunity, respond individually to unique glycoprotein markers known as antigens present on all cells. Right? We have the cell-mediated immunity brought about by two types of T cells, antibody-mediated immunity carried out by B cells. So adaptive immunity responds more slowly, usually takes 3 to 5 days. But after that point, it is the dominant response. That exposure to the antigen is remembered, right? And that's where we want to say adaptive immunity is remembered by specific lymphocytes and antibodies, allows a more rapid and efficient response if we ever see that enemy again. Adaptive immunity and innate immunity work together, right? They do not operate individually, they work together. But the first line of defense is the body is covering, our skin, mucous membrane. Both the skin and mucosa provide a physical barrier. Skin is resistant to mechanical stress. Remember, skin is waterproof. We have this hard protein called keratin. We have sebaceous glands which secrete sebum, which is slightly acidic, which keeps a lot of things from growing on our skin. We have mucous membranes, which line all passageways in the body that open to the outside. This sticky substance called mucus traps pathogens and other debris and protects underlying cells. We also have the stomach, which secretes acid, kills ingested pathogens. So that's our first line of defense. Then we have cells and proteins of innate and adaptive immunity. Main cells of the immune system are different types of leukocytes. So we have the agranulocytes, don't have the granules on the inside, the B and T lymphocytes and monocytes. We have granulocytes, neutrophils, eosinophils, basophils, right? And then we also have NK cells in the blood and spleen, dendritic cells, antibodies produced by the B lymphocytes, complement system, which are proteins. We have cytokines, also a diverse group of proteins secreted by cells, both innate and adaptive immunity, have various effects. The lymphatic and immune system work closely together, as we've already sort of hinted at. Lymphoid organs and tissues provide resistance for cells of the immune system. So the B cells, T cells, macrophages take up residence in the lymph nodes, in the malt, in the spleen. They live in these lymphoid organs, so they're readily available in these organs. The lymphoid organs and tissues trap pathogens. We have this reticular fiber produced by these reticular cells. So the pathogens, the foreigners, the invaders get trapped in this net, and then you have these macrophages, T cells that are already there that are ready to attack as soon as they get trapped. This is set up, and then we have lymphoid organs activate cells of the immune system. So some of these lymphoid organs can activate these cells of the immune system and get them activated so that they come and attack. So that it's all working together. All right, so let's talk about innate immunity. We have phagocytes. Phagocytic cells include macrophages, neutrophils, eosinophils. Remember, phagocytosis, cell eating, a cell ingesting something. And these phagocytes will ingest some kind of pathogen, bring it into the cell, and then destroy it. Macrophages. Activated local macrophages are generally first cell to respond to cellular injury, ingest other cells and cellular debris, kill it with hydrogen peroxide and hypochlorous acid, so whatever it's ingested is killed. Also have a cytotoxic effect, can secrete chemicals onto the pathogen, ones that are too large to ingest and kill them that way. T cells become activated by displayed antigens that macrophages can display, and in turn secrete substances that increase activity of macrophages. So they're usually the first to respond. Neutrophils are usually the most numerous granulocyte, highly effective, kill their ingested pathogens with chemicals such as hydrogen peroxide, hypochlorous acid, and lysozyme. They can ingest many types of cells, destroy them, generally reside in the blood, and must be recruited to damage tissue by chemical signals. Dendritic cells, like macrophages, function as most important antigen presenting cells. Substances they ingest are presented to T cells and also to B cells, which are then activated, right? And B cells and T cells help with the adaptive immunity, especially. We also have eosinophils, so phagocytes that can migrate from the blood tissue, primarily involved in parasitic pathogens. They can destroy them. We also have NK cells, natural killer cells, have remarkable ability to recognize cancerous cells and cells infected with certain viruses. Also cytotoxic, release substances that can destroy their target cells. All right, so all these cells work with immunity. Basophils, rare granulocytes, so they have the granules in the cells. Those granules contain chemicals that mediate inflammation. Located in the blood, particularly involved in allergic responses. And we're going to talk about those when we talk about allergies and allergens. Right. So besides the cells, we also have antimicrobial proteins, including complement proteins and several types of cytokines. So we have cells, macrophages, eosinophils, neutrophils, NK cells, basophils. Those are different types of cells that are in the body that migrate to a specific area when there is an issue, when there is an innate response, when there is an issue, pathogen, or cell damage somewhere. But we also have, as part of our innate response, not our non-specific response, we have complement proteins, antimicrobial proteins. So the complement group of molecules, collectively known as complement system, consists of 20 or more plasma proteins in the blood plasma produced primarily by the liver. Remember, there's proteins that exist in our blood plasma. Blood plasma primarily made of water, but it has those proteins that are floating in that fluid that we call plasma, which is a part of the blood. Remember, if you centrifuge blood, you have the red blood cells, the hematocrit. You have the plasma sort of at the top, and those proteins in the blood plasma. Remember, those also uh, play a huge role in the osmotic pressure of the blood. So start putting all that together. So these complement proteins are designated with C and a number, play a critical role in both innate and adaptive immunity. Right? The complement proteins circulate primarily in an active forms. So in the blood plasma, they're circulating in an inactive form. Three series of enzymatic reactions activate complement proteins. We have a classical pathway. Inactive complement proteins bind to antibodies bound to an antigen. A lectin pathway initiated when proteins called lectins bind to carbohydrates on surface of microbes. An alternative pathway. These inactive complement proteins that are in the blood plasma encounter foreign cells such as bacteria. Right? Two pathways converge at cleavage of inactive complement protein called C3 into an active form C3b, which in turn cleaves to an active protein C5, cleaves an active protein C5 into its active component C5b, right? And so we have these different ways to activate these complement proteins. Right? They get activated by these different pathways. And then what happens? Activated complement proteins lead to the following effects. Cell lysis. So some complement proteins that are floating around in your blood plasma become activated, and they're able to lyse or make, basically pop or blow up um, plasma membranes of pathogens. If a cell explodes, it's destroyed, right? Binds to the surface of the pathogens, provides docking site for several other activated complement proteins. Together, these complement proteins form a structure collectively known as membrane attack complex, MAC, creating pores that cause it to lyse. Complement proteins enhance the inflammation, the inflammatory response. So triggering basophils and mast cells to release chemicals that mediate inflammation. Complement proteins can neutralize viruses, attack the complex to bind to certain viruses and neutralize them, block them from affecting host cells. Right, so that's shows you the effect of some complement proteins that are floating around in your blood plasma right now. Attack a bacteria, calls it the lyse, and that once a bacteria lyses, it like blows up, it's dead. Complement proteins can cause more basophils. Complement proteins can neutralize viruses. We also can enhance phagocytosis, cell eating. So the C3b acts as an opsonin by binding to pathogens, makes phagocytes bind more strongly to the pathogen. So it basically makes that pathogen more likely to be ingested uh, by a phagocytic a cell that operates by phagocytosis. Remember, the macrophages operate by phagocytosis. So the complement protein can make it easier for the other cells to come and destroy this cell by phagocytosis. Remember, the macrophages can bring it in by phagocytosis and then use harmful chemicals like hydrochloric acid to destroy it. We also have the clearance of immune complexes. C3b binds to immune complexes, triggers their phagocytosis, um, and clears them from circulation. Okay, so the complement proteins bind to this bacteria. The macrophages more likely to ingest it, or the complement protein binds to this immune complex, this virus or something like that, and makes it more likely for the macrophage again to bring it in and destroy it. We also have cytokines, depending on how you want to say it, proteins produced by several types of immune cells, which enhance immune response. Right? We have tumor necrosis factor, cytokine secreted primarily by activated macrophages, response to certain bacteria and other pathogens. Attracts phagocytes to area of infection, increases phagocyte activity. Interferons, also produced by macrophages, dendritic cells, NK cells, inhibit viral replication. Interleukins, a protein produced mainly by various leukocytes, which stimulates production of neutrophils by bone marrow. We want to make more neutrophils, stimulate NK cells, trigger production of interferons, activate T cells. More of them that are made, the more that can come and fight. So the interleukins can help produce more leukocytes, bring more. Think of leukocytes, think of like soldiers, bring more soldiers to the fight. All right, so what is inflammation? We talk about, you've probably heard that word inflammation. Inflammatory response is the innate response, innate meaning non-specific response that occurs when a cell is damaged by anything. Could be trauma, could be bacteria or virus, toxins. You can get inflammation from heat. You can get an inflammatory response from chemicals. But basically, what is happening is damaged cells. So, whether they're damaged by heat, whether they're damaged by chemicals, whether they're damaged by trauma, damaged cells release inflammatory mediators that cause local changes in damaged tissue. The phagocytes, that's your initial line of defense, your macrophages, your neutrophils, that's your initial line of defense. Phagocytes arrive and clean up the damaged tissue. So, part one, release of inflammatory mediators, right? So, tissue damage happens by one of those methods. We release these inflammatory mediators, histamine, serotonin, cytokines, prostaglandins, activated complement proteins. Remember, they're floating around in your blood plasma, trigger the release of the inflammatory mediators from cells such as basophils and mast cells. Injured area becomes red and swollen, feels warm to the touch, and it hurts. Your body is working right there, right? Redness, heat, swelling, and pain. So why does that happen? Inflammation, right? So one of the things that happens is vasodilation. So cells get injured, they release inflammatory mediators, vasodilation occurs. So histamine that's released, bradykinin, vasodilation, dilate to get bigger, vasodilation. So those blood vessels get bigger. And there, so smooth muscle and the arteriole supplying damaged tissue allows more blood to come to that area. Redness, blood gets your redness. More blood to that area also brings heat. Think about it. But blood is your same temperature as your core temperature, 98.6 degrees. If an area of my body is signaling for more blood, it's going to turn red because blood is red. Blood is being pumped there. That arterial, that systemic blood is being pumped there. And then it's also going to be warm because it's going to be 98.6 degrees. So that's how we get that redness and heat. We have increased capillary permeability, right? So protein-rich fluid to leak from the blood vessels into the tissue spaces also will cause swelling. So the redness, more blood, heat, more blood, more fluid leaking from the blood, your proteins that are in your blood plasma leaking into that interstitial space, into the tissue, the space between the cells and the tissue, swelling, edema. Proteins in fluid include clotting proteins such as fibrinogen, complement proteins. Remember, those complement proteins can um, start that immune response. What about pain? We have redness, we have heat, we have swelling. But what about pain? Why do we have pain when there's an inflammatory response? So several inflammatory mediators trigger action potentials. Let us know when our tissues are being damaged to avoid further damage. So these so action potentials are being triggered, can cause temporary loss of function, also allow the body to begin to repair itself, right? We don't want to use that part of your body. You kind of want it to stay still so that it can start to repair. And we also have the recruitment of other cells. Inflammatory mediators recruit leukocytes, right? So when these inflammatory mediators go out, we get those white blood cells coming in. Macrophages, neutrophils. Remember, macrophages are the first ones to come in. Complement proteins are being activated. Clean up the area. Remove the damaged cells. As the phagocytes get there, macrophages, for instance, they enlarge, begin to phagocytize pathogens, damaged cells. Neutrophils come to damage tissue. Inflammatory mediators activated, complement proteins, attract neutrophils, enable them to leave blood and enter tissue, increase capillary permeability. Bone marrow releases stored neutrophils in the blood. They're there to help out. Cytokines produce activated phagocytes, act on cells in bone marrow to increase production of neutrophils and monocytes. So you get more and more of these phagocytic cells coming. Elevated number of leukocytes allow damaged area to be cleared and pathogens removed so that we have cells such as fibroblasts can begin the healing process. The accumulation of these dead leukocytes is what we know of as pus. So when pus fills a wound, that's dead white blood cells have done their job. Soldiers dying on the battlefield, and they form what is called pus. And that's our inflammatory response. Now, there's a lot of medications. All of these things are natural when we have inflammatory response. Redness, swelling, uh, heat, pain. All these things are natural when we have an inflammatory response. That's your body working to fix whatever is going wrong. But there are a lot of drugs that work to try to minimize the inflammatory response. People don't want to be in pain. And so we try to block the production of prostaglandins, for instance, uh, and to try to reduce the amount of pain, right? Ibuprofen, reduce the amount of pain. Doesn't necessarily heal the body any faster, but it can do things like reduce the pain. A fever, temperature that is above the normal range, is usually the sign of an inflammatory response, inflammatory process somewhere in the body. It is part of our innate response, and happens when chemicals called pyrogens are released from damaged cells or certain bacteria. They act on the hypothalamus, which regulates many aspects of homeostasis. Our body gets sort of tricked, and our hypothalamic thermostat resets to a higher range, and therefore we feel like we're supposed to be uh, that our normal body temperature is too low, and so we feel this sense of coldness in our normal body temperature, and we get what is called the chills. But this is just all part of a inflammatory response. Your body is fighting off something. This is the summary of our innate immunity, our initial immunity. And then we have what is called adaptive immunity. Right? So cell-mediated immunity involves helper T cells, cytotoxic T cells. These cells respond primarily to cells infected with intracellular pathogens, cancer cells, and foreign cells. T cells are formed in the bone marrow. They leave the bone marrow and mature in your thymus. Remember that as we age, our thymus becomes less functional. Populations of T cells can respond to specific antigens. Those populations that respond to a specific antigen are known as clones. There are millions of types of clones. Only a few cells of each clone exist in the body. Your thymus screens which ones are going to respond to clones and which ones are not. The ones that are not are going to be destroyed so that your body is ready for a response to foreign antigens. Right? So that's the process of that's shows you how we get T cells created in the bone marrow, mature in the thymus, and then they reside a lot of times in the lymph nodes. An antigen is a substance that a B or T cells recognize. It comes from the phrase antibody generating. It's usually a peptide, but could be a carbohydrate or a lipid or metal. Immunogens, a certain type of antigen, are capable of generating response from the immune system. Antigens present on your own cells are called self-antigens and are not immunogens. You have haptens, very small antigens, which are immunogenic only if attached to protein carriers. And highlighted here, I have T cell receptors are found on the surface of every T cell. Receptor must bind specific antigen before cell can become activated. Right? So we have this T cell bind a receptor must bind specific antigen before a cell can become activated, right? And that's what's going to give us this this learned response. T cells can only interact with pieces of the antigen bound through a glycoprotein called a major histocompatibility complex, MHC molecule. So the MHC molecules are found on nearly all nucleated cells, except blood cells, that's why blood cells are easier to donate. But the MHC molecules serve as a docking site for specific components of antigens that are then displayed to T cells. Right? So we have this MHC molecule, which works with the antigen, then it's displayed to the T cell, and the T cell can bind to that. The T cell that is a clone, that is a specific for a different type, for each specific type of pathogen. There are two types of MHC molecules. We have class 1 on the plasma membrane surface of all nucleated cells. They present endogenous antigens, cytotoxic T cells. Apologize. Kids coming in the room while I'm recording the video. So we have these class 1 MHC molecules, which are on the plasma membrane surface of nearly all nucleated cells. And then you have these class 2 molecules only on surfaces of antigen presenting cells, right? So this can get a little confusing. The class 2 molecules, helper T cells, generally interact with the class 2. Cytotoxic T cells generally interact only with class 1. Right? Class 1 endogenous present endogenous antigens synthesized inside the cell. Class 2 exogenous antigens taken in by phagocytosis. So exogenous antigens originate outside the cell. The cell must take it in, cell eating, phagocytosis. Endogenous antigens originate inside the cell, but can originate different ways. So foreign antigen present on a pathogen that lives inside your cell, foreign or self-antigen encoded by your DNA can make an endogenous antigen. Endogenous antigens can be foreign or self, but exogenous antigens are nearly always foreign. All right, so basic steps by which class 1 MHC molecule processes, displays endogenous antigen. Cell synthesizes either self-antigen or foreign antigen. Antigen is broken down by enzymes in the cytosol. Remember, that's in the cell, the fluid in the cell. Antigen fragment containing antigenic determinant transported into the RER, referring to plasma reticulum, coupled with the class 1 MHC. MHC antigen complex leave RER by vesicle, and then it's presented on the cell's plasma membrane. Right, so that's the process of an antigen processing with the MHC, class 1 MHC molecule. It presents on the surface, which can be recognized by a T cell. The other one, the exogenous antigens, class 2 MHC molecules, cell phagocytoses the pathogen, phagocytic vesicle fuses with lysosome, pathogen is degraded, antigen fragmented, lysosome fuses with vesicle from RER, rough endoplasmic reticulum, that contains class 2 MHC, binds to MHC molecule, MHC antigen complex is inserted into the cell's plasma membrane. And that's how the class 2 exogenous antigen is displayed on the surface so that the T cell can recognize it. The process of a naive T cell activation is very similar for helper T or cytotoxic T cells. And the main difference is the type of MHC molecule. Dendritic cells have unique ability to undergo cross-presentation, able to display some of ingested viral and bacterial antigens on both class 1 and class 2 molecules, enables them to interact with and activate naive cytotoxic T cells as well as naive helper T cells. Right? So what is the process? Dendritic cells display antigen fragments on their MHC molecules. The MHC antigen complex binds to receptor of specific naive helper T or cytotoxic cell clone. Dendritic cells process and display the antigen fragments, presents the antigen to T cell, specific for its individual MHC antigen complex. T cell receptor recognizes, binds this complex, what is known as clonal selection because antigen selects particular T cell clone. The helper T cell or the cytotoxic T cell binds the co-stimulator and becomes activated. The activated helper T or cytotoxic T cell proliferates, differentiates into effector cells and memory T cells. This is where we get that memory response. Those memory T cells are responsible for cell-mediated immunological memory, right? So the next time, respond more quickly and efficiently to subsequent exposures of this particular antigen, and there's no need for a co-stimulator, and it's much more faster. All right, so that's showing you the how that's happening. What effects does the T cell have? Well, the helper T cells and the cytotoxic T cells have different roles. The helper T cells have no phagocytic or cytotoxic abilities. Term helper T. They exert their effects through the secretion of cytokines, activate and enhance various components of their immune response. So these helper T cells are required for normal function of all components of your immune system. That's why things like HIV, which mess up the function of our helper T cells, can cause such problems, right? So our helper T cells work with our innate, our initial response, as well as our antibody-mediated and cell-mediated immunity, right? So helper T cells secretes cytokine interleukin-3. Remember that stimulates macrophages to become more efficient in phagocytosis. Also causes macrophages to produce interleukin-12, stimulates helper T cells to generate more interleukin-3 and positive feedback. Right, so they sort of stimulate each other. More effects of the T cells. We have adapted cell-mediated immunity, activation of cytotoxic cells, so that those helper T cells, when they secrete the cytokine interleukin-2, that is required to activate the cytotoxic T cells. They also stimulate B cells. So helper T cells directly bind to B cells to stimulate them to proliferate and differentiate. Also secrete various interleukins that stimulate B cell proliferation and increase antibody production, which we're going to talk about more when we talk about the B cells. Right? The cytotoxic T cells, their job is to kill, right? They're fighters. All right, so they specifically work with foreign antigens bound to class 1 MHC molecules. They can detect abnormalities in any cell types with a nucleus, cancer cells, foreign cells, right? They can seek. They're like private detectives, finding the enemy. They're activated very similar to helper T cells, with the addition that they require the IL-2, the interleukin-2 from T cells in order to fully activate. Protective mechanism prevents the cytotoxic T cells from activating. But once they activate, they bind to the target cell, release perforin, um, which drills pores in the target cell's plasma membrane, um, and then release enzymes that now enter the target cell cytosol, degrade the target cell, and cause it to die. Cell death, cell apoptosis. So that's what the cytotoxic T cell wants to do, destroy the target cell that's not supposed to be in the body. A lot of times we have issues with organ and tissue transplantation. Autografts are trans when you transplant tissue from one part of your body to another part of your body, the same person's body. That usually doesn't have any problem. Isografts involve organ and tissue transplanted between two genetically identical individuals. Usually also does not present much of a problem. But allografts, which are most common type of graft, involve organs and tissues transplanted between non-identical individuals. And so your body can start to recognize, can sometimes recognize that graft or as that organ or that tissue as foreign and try to attack it. A rejected organ will get attacked because the cytotoxic T cells will try to destroy it. And that can lead to necrosis, and it can lead to death because it can lead to organ failure. So this is prevented by trying to suppress the immune system, and also trying to make sure people are as compatible as possible. All right, so now we go into the antibody-mediated immunity. This involves B cells and antibodies. So B cells have B cell receptors that bind to specific antigens. Group of B cells that bind to specific antigen is known as a clone, similar to the T cells. Antibodies secreted by B cell clone bind to the same antigen as B cell receptor. This antibody-mediated immune response has three basic phases. B cell clone recognizes its antigen, triggers it to change and secrete antibodies. The antibody level in the blood rises, directly responsible for actions that lead to destruction of antigens to which they bind. The persistence of this population of B cells react much more rapidly the next time around. We have this memory response. All right, so first phase, B cells develop and mature within bone marrow. Once again, similar to T cells, who start in the bone marrow and eventually go to the thymus, B cells develop and mature in the bone marrow and the lymphoid cell line. Billions of B cells are produced each day. Only 10% finish the maturation process. B cell clones that complete maturation enter circulation, eventually take up residence in the lymphoid organs, spleen, lymph nodes. Remember that lymphatic system and the immune system work closely together. When antigens enter the body, they're captured in these lymphoid organs. B cells that are not exposed to the specific antigens within a few days to a few weeks will die. That's the process of B cells, the bone marrow, to the lymph node. Naive B cells that do not encounter antigens become activated by this process. So B cell clone binds its antigen and is activated. Clonal selection is when antigen binds to B cell receptor on surface of specific B cell clone. A sensitized B cell processes the antigen surface of some type of cell and presents it on its class 2 MAC molecules. B cell then binds to the helper T cell to become fully activated. Starts a whole series of events inside the cell that triggers transcription, antibody genes, and multiple other changes that activate the B cell. B cell divides repeatedly, resulting cells differentiate into plasma cells, which secrete what we know as antibodies, and also memory B cells. Long-lived cells do not secrete antibodies, but will increase that response quickly. Dendritic cells and other APCs continue to expose plasma cells and other B cells to antigen. All right, so that's your response. See how the helper T cell helps this process. What is the effect of antibodies? All right. So the second phase of antibody-mediated immune response involves antibodies and their effects. So serology is the study of antibodies, studies their structure, functions, how they work in medicine research. The basic unit of antibodies is Y-shaped molecule, 4 peptide chains, 2 heavy, 2 light. You have this constant C region, which is relatively similar, and you have this variable V region, which is a unique sequence of amino acids responsible for antigen recognition and binding. Right, so we have the constant region, which is basically the same for most antibodies, and then we have this variable region, right, to make our antibodies specific. Antibody has V regions at tips of arms of molecules, so basic subunit of an antibody has 2 antigen binding sites. You have different monomers, dimers, pentamers, depending on how many subunits are bound together. 5 basic classes of antibodies, right? So we have 2-letter abbreviations for antibodies: IG, immunoglobulin, followed by its designated class. Remember, there's 5 basic classes. We have G, A, M, E, and D. IgG is most prevalent, single subunit, only antibody able to cross from blood of pregnant women to her developing fetus through the placenta. A, IgA, usually a dimer, 2 subunits, present in secretions from skin, mucous membranes, exocrine glands. M is a pentamer, 5 subunits, generally first antibody secreted by plasma cells. The body is invaded by a pathogen. E, single subunit antibody, very low amounts, binds 2 types of antigen, parasitic pathogens such as tapeworms, allergens, environmental antigens. D, unique because it's the only antibody not secreted by B cells in significant amounts. Single subunit located on surface of B cells. Similar helps activate B cells similar to IgM. So antibodies in a particular class feature similar C regions, but each antibody has unique V regions. So they have a constant region similar, but their V region is different, their variable region. There's remarkable variability in antibodies V region, 1 million or more different antibodies in the body at a time. So it's capable of recognizing all these different antigens. The B cell clone that starts out producing certain class of antibodies, such as IgM, has the ability to switch to production of different classes, such as IgG. The mechanism for this process involves genetic rearrangements to yield different antibody C regions while maintaining same V region. In this way, antibodies can have different effects while binding same type of antigen, right? So multiple ways we can create these antibodies. Different antibodies, which will bind to different antigens. And then, so what's the effect of these antibodies? Well, these antibodies, which are secreted, so actions of antibodies are based on their ability to bind antigens, lead to multiple effects. Agglutination, that's a very good effect. So, antibody binds to an antigen on one or more cell, on more than one cell, binds to multiple antigens on multiple cells. So multiple antigens on more than one cell, and clumps. Those cells create a big, nice clump now, um, cross-linked by their antibodies, and we have agglutination. Precipitation, similar to agglutination, involves soluble antigens instead of whole cells. Both decrease the solubility of that antigen antibody complex, so they make them clump up and make it easier for phagocytes to ingest the complex. IgM is most potent in agglutinating and precipitating antibody due to its 10 antigen binding sites. Antigens can also operate in opsonization, involves molecules such as complement and IgG antibodies. They can coat the pathogens, bind, and activate phagocytosis. All right, so make them easier to be ingested by macrophages, for instance. Antibodies can neutralize and prevent them from interacting with our cells. Antibodies, IgM and IgG, bind to and activate complement proteins of innate immunity. So those proteins that are floating around in your blood plasma, they can bind to those and help activate those. So our innate working with our learned immunity. Antibodies can stimulate inflammation. IgE directly triggers inflammation, initiating release of inflammatory mediators from mast cells and basophils. Antibodies trigger inflammation. Inflammation causes our leukocytes, other cells to come and get busy. But most antibodies don't directly kill pathogens. They either render them less harmful or make them more able to be destroyed by phagocytosis or the complement proteins. The memory B cells are responsible for the antibody-mediated immunological memory. These B cells respond more efficiently when antigen is encountered. Second time, which is the third phase of our immune response. So, upon first exposure to an antigen on the surface of a cell that's not supposed to be in our body, the B cell clone specific for that antigen recognizes it, proliferates, differentiates into plasma and memory B cells, and plasma cells begin to secrete antibodies. And this response is called the primary immune response. Right? This is a slow response, 4 to 5 day response as B cells proliferate, differentiate into these plasma cells and memory cells, and begin to secrete antibodies in response to this specific antigen. The antibody levels peak around 7 to 14 days, and it's during this lag phase that you feel sick. But in the future, when you see that same antigen on the surface of that cell, it results in the activation of those memory B cells, which were formed the first time this pathogen, this cell that didn't belong into your body was in the body. So now the second time, or third time, or fourth time, or fifth time, when these memory B cells encounter this antigen, for which they are specific, we have the secondary immune response, lasts longer than the primary response, and happens quicker. So the lag phase is only 1 to 3 days. and its antibody levels peak more rapidly at 3 to 5 days and reach a peak 100 to 1,000 times larger. So much more antibodies are made, which can bind to and cause agglutination, which can trigger the inflammatory response, which can cause the fight to happen even quicker the next go round. The major antibody involved in the secondary response, IgG, whereas it's IgM in the primary response. Immunological memory. So we have that first level, our skin and our mucus. We have that second level, the innate, the macrophages, the eosinophils, the neutrophils, the um, complement proteins, um, the lymph nodes, the spleen, the sort of our innate response. And then we have the third phase, which are our adaptive response, which is created by the antibodies, our and our T cells and our B cells. Vaccination, also known as immunization, involves exposing individuals to an antigen so that they can get the response, develop the memory B cells for that specific antigen, so that the next time that antigen, or when they do see that antigen again in quote unquote real life, you already have the antibodies made for it. You'll go right into your secondary immune response instead of your primary immune response. And that is the big picture. There can be many disorders to the immune system: hypersensitivity, immunodeficiency. Hypersensitivity, your body's overreacting to damage. Immunodeficiency, some part of your immune system is failing. Autoimmune, your body's fighting itself, right? So hypersensitivity, different types. Most common is allergies. The body reacts to a foreign antigen, allergen, and then it sort of overreacts to that, triggers vasodilation, increased capillary permeability, smooth muscle spasms. Asthma is an allergic respiratory response. It's hypersensitivity, something that's not that bad for you, but your body responds like it's terrible. You may have heard of anaphylactic shock, which is a dramatic, immediate hypersensitivity reaction, something that is not going to harm you, but your body acts like it's dying, basically, and has this severe reaction. And so it's like your body basically hurting itself because it's overreacting to uh, whatever it is. And this can happen. People have nut allergies and they can go into anaphylactic shock due to how their body over responds to it. Swelling, blood pressure dropping, all kinds of things can happen. And treatment is epinephrine is one way to treat it. Right? There's also immunodeficiency disorders. AIDS is one of those, acquired immunodeficiency syndrome, where it basically causes your immune system not to function properly. AIDS is caused by the human immunodeficiency virus, HIV-1. It's a retrovirus, operates by reverse transcriptase, basically causes your body not to function properly. The T cells do not function properly in your body. And then what ends up happening with things like AIDS is you end up dying from diseases that you shouldn't die from because your immune system is just not functioning properly. And then we have autoimmune disorders. So T cells, B cells secrete antibodies that bind to self antigens. Remember, cells in your body have antigens that are good cells. And when we have T cells and B cells that have antibodies that bind to antigens on good cells, your body starts attacking itself. So, for instance, multiple sclerosis is an example of that. The body attacking itself. And that is the end.